How cryotherapy preservation techniques could increase the viability and motility of Apis mellifera drone sperm. Reproduction for Apis mellifera occurs between the queen and drone bees. In the springtime every year, drones leave the hive and swarm about 1.5 kilometers away from the hive. Queens will then take their mating flight lasting from 5 to 30 plus minutes. They typically take just one flight a day and mate with multiple drones. Once mating has occurred, unfortunately for the drones, their endophallus breaks off in the queen and they fall to their death. The mature sperm then make their way into the queen's spermatheca, in which they are naturally diluted. The sperm can stay alive and viable for several years inside the spermatheca, although many bees do not have that long of a lifespan. In recent years, the bee population has been at a rapid decline due to human development and other environmental factors. Scientists have been looking into ways to preserve the bee population. Cryotherapy has been one of those techniques. So what is cryotherapy? Cryo means cold or freezing, and therapy is synonymous to treatment. Although cryotherapy is most known for its effects on humans, in recent research, cryotherapy has been used in the preservation of honeybee sperm. It may seem like a strange technique, but because of recent decline in bee population and diversity, scientists have been looking for ways to increase the lifespan of a drone sperm. So you may be asking yourself, how do cryotherapy techniques compare to normal reproduction in terms of the motility and viability of Apis mellifera drone sperm? As I mentioned earlier, the sperm are naturally diluted with a balanced salt solution or diluent in the queen's spermatheca. This dilution increases the longevity of sperm. I bring up this aspect of the reproductive system of Apis mellifera because in many studies regarding cryotherapy, scientists have to artificially dilute the sperm. A common diluent used in lab is called DMSO, or dimethyl sulfoxide. In many of the studies, this diluent is used to imitate the natural dilution that occurs in the spermatheca. Looking at the diagram to the right, you can see the DMSO, or non-activating dilution, has a higher proportional sperm viability than other dilutants and even the control group. Knowing which diluent works best to recreate natural dilution that occurs in the queen has helped scientists looking into cryotherapy to know how best to imitate the mating process. Once the experimenter has decided to use diluent or not, the type of cryotherapy can also differ. The most popular methodology right now is called slow programmable freezing. This technique is prefaced with a slow cooling phase where semen is chilled slowly to 4 degrees Celsius for 2 hours to prevent a cold shock. The semen is frozen to -40 degrees Celsius at a rate of -3 degrees Celsius per minute before it is directly plunged into liquid nitrogen to be stored indefinitely. Another technique is vitrification, or flash freezing. Vitrification is different in that the samples are directly plunged into the liquid nitrogen. By directly exposing samples to liquid nitrogen, cells transform into a glass-like state and prevent ice formation entirely. Such a transition requires a freezing rate of at least -1,000 degrees Celsius per minute. A final technique is vapor emission. Vapor emission exposes samples to nitrogen vapor produced as liquid nitrogen evaporates. This method is less common than vitrification or slow programmable freezing. It has been successful for human sperm cryotherapy, but less successful when applied to honeybee sperm. Vapor emission freezes samples faster than slow programmable freezing, but slower than direct liquid nitrogen exposure. Looking at the graph, you can see all 3 techniques used. For clarification, cryolock and dialysis are just 2 devices of freezing. Looking at the results, we can see that the cryotherapy techniques, slow programmable freezing gives the highest results in regards to the motility of the sperm. Now, all of that being said, many experiments are still finding that cryotherapy sperm are not as motile or viable as fresh sperm. Looking at this graph and thinking back to the graph on the previous slide, you can see the control groups perform better in terms of motility and viability. So although cryotherapy is an alternative, it is not as effective as normal reproduction. To sum all this up, cryotherapy is a relatively new thing for the honeybees. It has been used for humans most prevalently in recent years, and it could be something really useful for the preservation of honeybee sperm. Research has shown some positive results regarding the motility and viability of sperm with the use of cryotherapy, but ultimately not enough experiments have been done to perfect the artificial reproduction process in a lab. Cryotherapy, although not showing as efficient results as the control group, with a little more research could be the new way to save the bees. There is an urgent need for cryopreservation of honeybee sperm. The frightening losses of honeybee population and humans' vital link to the species means the ability to properly preserve and distribute honeybee genetics is now crucial. Honeybee biodiversity is currently under a massive threat from the introduction of foreign genes into native populations. Cryobanking of bee semen could be the newest tool to help preserve bee diversity.